This lecture covers some microbial diseases of the circulatory and lymphatic systems. Our circulatory system and lymphatic systems are networks of vessels that are all throughout our body. And then we have this pump, our heart, that transports blood and lymph throughout the body and helps the movement of those systems. So you can see whenever these systems become infected with a microorganism, they're suddenly given a fast-track ticket to go nearly anywhere in our body. And so these vessels can facilitate this rapid dissemination of microorganisms to other regions of the body. And this can potentially be very serious and lead to a much more exaggerated immune response and a more serious infection. First off, the anatomy of the circulatory system. So the whole goal of the circulatory system is to move blood throughout your body. So you want to deliver nutrients to tissues, you want to deliver immune factors, you want to deliver oxygen, and you want to take away waste products. Because specifically talking about us, remember, we are obligate aerobes. We need oxygen to get energy, and our blood is delivering oxygen and then taking away deoxygenated blood so we can breathe out carbon dioxide. And all this is being pumped throughout the body. The circulatory system, the circulation is happening because of a pump called our heart. So our heart is a 4-chambered pump that propels blood throughout the body. And then we have other important organs for our circulatory system as well, like our kidneys, liver, and spleen that help with the filtering of our blood. The anatomy of the lymphatic system. So the goal of the lymphatic system is to protect our body from illness. This is where a lot of white blood cells hang out, a lot of lymphocytes hang out, B cells, T cells, natural killer cells, other white blood cells that are surveying your body for potential invaders. The lymphatic system also maintains our body fluid levels and removes cellular waste, so you can think of it as your body's sewage system. And unlike the circulatory system, the lymphatic system is not a circulating system. So it picks up fluids from parts of the body and then dumps it or empties it at veins near the heart. So it kind of picks up this waste and extra fluid, filters out that waste, and then dumps near the heart. So it is not constantly circulating. There are primary and secondary lymphoid tissues. So your primary lymphoid tissues would be things like your bone marrow, where all of your white blood cells and red blood cells and all of those things are made. Also like your thymus, that is a primary lymphoid tissue. Then secondary lymphoid tissues like your lymph nodes and your lymph vessels. Now, infections of the circulatory system, just kind of a general overview. So our circulatory system, our blood, should be sterile. There should be no normal microbiota in your blood. And our circulatory system is a closed system. So there's no easy portal of entry for a microbe. One, it's a closed system because it's transferring oxygenated blood and then coming back deoxygenated to the heart, becoming oxygenated. Yes, it's closed that way, but you can also think of it as kind of closed within you. So your blood vessels aren't just open to the environment. Kind of like our skin is constantly in contact with the environment. So how would a microbe get into this closed system, and what kind of immune defenses do microbes have to overcome in order to infect this system? Well, one, to get into this closed system, it would have to be able to get access most often by some type of open wound. So when you have an open wound, a pathogen has direct access to your circulatory system at that point. So it's past that first line of defense of your skin and mucous membranes. And then immune defenses, we talked about a lot of different immune defenses that are just hanging out in our circulatory system and lymphatic system. You have a whole array of white blood cells and complement proteins that are constantly surveying your body for potential invaders. So there you have a lot of different immune defenses at the ready in your circulatory system to prevent pathogens from entering that system. And they're constantly surveying because there should be no normal microbiota in that circulatory system. So some important terms to kind of know in describing conditions involving microbes in the circulatory system once they do get there. Bacteremia refers to bacteria in the blood. Septicemia refers to bacteria that are reproducing in the blood as they spread. So septicemia is more kind of dangerous than bacteremia. Septicemia involves bacteria that are actively reproducing. Viremia is the presence of viruses in the blood, and then toxemia is the presence of microbial toxins in the circulatory system. And then we'll talk more about this in a little bit. In like a few slides, we'll talk a little bit more about sepsis. But sepsis is referring to the actual introduction of bacteria into your circulatory system, and then that bacteria actively replicating. And there are different levels of sepsis. And sepsis is really this kind of system-wide shock due to all of that bacteria in the blood and your immune system definitely having that heightened immune response because of the presence of so many pathogens kind of everywhere at once in your body. And ultimately, what this can lead to is something called septic shock. And septic shock is the most dangerous kind of last phase of sepsis, or this actively replicating bacteria in your blood, because during septic shock you have organ failure. And this organ failure is due to it's caused by this low blood pressure. Because now you have bacteria almost everywhere in your blood. Your immune system is like, oh my gosh, I'm panicking. So now you have kind of a system-wide inflammatory response. One of the things that happens during the inflammatory response is your blood vessels get wider to allow more blood to get to certain areas of your body. But now that all of your blood vessels have dilated, now your blood pressure has plummeted. And that means that blood isn't being effectively pumped around your body. That means oxygen is not being delivered to those tissues. That means it can lead to organ failure, which is the most dangerous and deadly phase. That is what is called septic shock. And then some important things about infections of the lymphatic system. So first off, actually, just keep in mind that these two systems are closely related to each other. They're both systems of vessels that are just extensive throughout your whole body. And then like the circulatory system, the lymphatic system has no normal microbiota, but it is constantly surveying for potential pathogens or invaders in your body. So there are a lot of immune cells typically present in your lymphatic system, just kind of hanging out throughout the lymphatic vessels or hanging out at lymph nodes, and then checking to see if there are pathogens present. And then they are going to eliminate microbes before they establish an infection or move somewhere else in the body. So obviously, if a microbe were going to cause an infection in the lymphatic system, it would need a lot of special virulence factors to overcome all of those defenses. Because in your blood and your lymphatic system, you have a lot of immune system cells constantly surveying for potential invaders. So anything that is going to cause an infection here must have some very serious virulence factors that allow it to evade your immune system. Here's some terminology for the lymphatic system infections. Lymphangitis, this is inflammation of lymphatic vessels, and you can often see it as one of the cardinal signs of inflammation. Redness, you can see an inflamed lymphatic vessel below the skin as this red line. And then lymphadenitis is the swelling of lymph nodes, and these swollen lymph nodes are also referred to as buboes. So that is interchangeable. A swollen lymph node or lymphadenitis is known as buboes. And this will come up again in a little bit because you may not realize it now, but bubo should sound a little familiar to you because you've probably heard of the bubonic plague. And that's where it gets its name from, is the development of those buboes or swollen lymph nodes as a result of that disease. Now we'll take the next few slides to talk a little bit more about bacterial sepsis, or bacteria in the blood. So bacteria are going to be the most common pathogens that lead to sepsis and potentially septic shock. So specifically, bacterial pneumonia is going to result in about half of those cases that it eventually can evolve and lead to septic shock caused by that initial bacterial pneumonia infection. And then most commonly, that infection is going to be caused by types of Staphylococcus, Streptococcus, and Pseudomonas. So this type of bacteria can initially cause a local infection, like with pneumonia, a local infection of the lungs, and that local infection can disseminate, if it is not treated, disseminate to other parts of the body, get into the bloodstream, and at that point it is bacterial sepsis. Septic shock is what I talked about on a few slides ago, and this is kind of the most severe stage of sepsis. So whenever you have bacteria in the blood and it's replicating, because septic shock results in that dramatic decrease of blood pressure, and this leads to organ failure and your organs not receiving enough oxygen. And this is from that kind of system-wide inflammatory response and your blood vessels dilating and your immune system having this exaggerated response to this system-wide bacterial infection. Next up, we have toxic shock. And so infections caused by Staphylococcus aureus can cause staphylococcal toxic shock syndrome, so TSS, and you've probably all heard of toxic shock syndrome before. And this is a result of Staphylococcus aureus having this very specific superantigen. This is called the toxic shock syndrome toxin 1, TSST1. So not all types of Staphylococcus aureus have this, but specific types of Staphylococcus aureus have this superantigen that can then induce this toxic shock syndrome. People that are at highest risk for developing staphylococcal toxic shock syndrome are going to be women that are already colonized by Staphylococcus aureus. So Staphylococcus aureus can potentially be part of the normal vaginal microbial community, and then when that microbial community experiences dysbiosis, maybe from taking some type of drug or from leaving in tampons too long or devices in the vagina too long, then that can lead to an upset in that microbial community and allow Staphylococcus aureus to proliferate, and that can potentially lead to the infection of Staphylococcus aureus proliferation and ultimately the disease. These are not the only people, though, that are at risk for toxic shock syndrome. Anyone can potentially get it. That is just the most at-risk population. Treatment of staphylococcal toxic shock syndrome includes debridement, so removing, it's kind of scraping and removing tissue. It can also be chemical debridement by giving local drugs to kind of clear infected tissue. Also elevating blood pressure, so preventing that shock and not allowing your blood vessels to kind of all dilate. So constricting blood vessels and elevating the blood pressure so your tissues can get oxygen. And also antibiotic therapy to decrease the population of Staphylococcus aureus. A very similar type of syndrome is caused by Streptococcus pyogenes instead of Staphylococcus aureus, and this causes streptococcal toxic shock syndrome. And this is a lot like toxic shock syndrome, but it's caused by a different bacterial species, and it's actually a lot more deadly than toxic shock syndrome caused by Staphylococcus aureus. A couple more examples of bacterial sepsis, bacteria in the blood that can also be caused by injury or medical intervention. So first up here we have puerperal sepsis. So this is also called childbed fever, and this is a nosocomial infection. So it's occurring in the hospital. It's associated with hospitals. And this occurs right after childbirth, that period of time when a woman is very exposed and more likely to experience an infection. So this is most often going to be caused by the bacteria Streptococcus pyogenes, but it can be caused by other types of bacteria as well. And puerperal sepsis can be greatly reduced just by adhering to sanitary protocols, like hand washing and aseptic techniques during childbirth. Then we have infectious arthritis. This is also called septic arthritis, and this is acute or chronic inflammation of specifically joint tissue caused by bacterial pathogens that are infecting that specific joint tissue site. So this can be the result of some type of trauma or injury of a joint that has been damaged, or from a surgical site. So this could also potentially be a nosocomial infection as well. And then we have bacterial endocarditis. This is infection of the tissue layer that lines the muscles and valves of your heart. And so people with pre-existing heart damage or people that have prosthetic valves in their heart are at higher risk for developing endocarditis because your heart is already dealing with kind of other things. The immune system is already very aware that there is something foreign there. And then these diseases can be really rapid. It can destroy heart valves if left untreated. and it can lead to death in just a few days. At other times, this can take years to develop over time. And I'll show you on the next slide kind of what happens during bacterial endocarditis and how bacteria that do infect these parts of the heart develop these bacterial vegetations that make it a lot more difficult for the heart to do its job. This image is showing an example of what happens during bacterial endocarditis and the development and infection of these bacterial vegetations on these heart valves. And this, you can easily see here, these bacterial vegetations kind of reduce the ability of these heart valves to close and open properly, and the heart needs to be able to do that to provide the pressure necessary to pump the blood into the lungs or out the aorta to the rest of the body. Bacteria can also end up in the blood through zoonotic diseases, so it can be transmitted from animals to humans. Our example of a zoonotic disease that leads to bacterial sepsis is cat scratch disease, and this is a bacterial infection that can be introduced to lymph nodes by the scratch or bite of a cat. So the scratch or bite of that cat is breaking that first line of defense, your skin, and introducing this bacteria to that lymphatic system, to all of those vessels that can then go to lots of different places in your body. This infection is caused by the bacteria Bartonella henselae, and it actually infects red blood cells. It has these specific adhesion virulence factors that allow it to specifically adhere to red blood cells. And it has some very general signs and symptoms like fevers and chills and fatigue, and so it's kind of more difficult to diagnose. And then most often, this infection is going to be very mild. It's going to be self-limiting. You're going to get better on your own, especially if you are not immunocompromised. The best way to prevent this infection is just by keeping cats free of fleas. So fleas are actually what is introducing this bacteria to the cats. So cats are scratching themselves or scratching fleas. They're cleaning their paws and everything, and then they're getting that bacteria into their mouth or under their claws, and then when they scratch or bite you, they're transmitting the bacteria. But it's coming from this kind of intermediate vector, the flea. And you can also prevent this after getting scratched or bite by a cat. Just wash with soap and warm water immediately. Another example of a zoonotic disease, or a disease that's transmitted from animals to humans that can lead to bacterial sepsis or bacteria in the blood, is the plague. And this is caused by the bacteria Yersinia pestis. And this has a pretty high mortality rate when it's left untreated. And we'll talk about 3 different forms of plague. So there's bubonic plague, pneumonic plague, and septicemic plague. Bubonic plague is going to be the most common form of the plague, and that's over 80% of cases are considered bubonic plague, and that's the result of this bacteria Yersinia pestis being transmitted by the bite of an infected flea. So the flea is responsible for transmitting this bacteria to humans. It can also be transmitted through intermediate vectors like mice and rats that are infected with these fleas. So that is the most common, and it's the has the lowest mortality rate. So people that have bubonic plague, if completely left untreated, it can have it has a mortality rate of, I believe, just somewhere from like 1 to maybe 10%. But there is treatment available, and with treatment, all of these are survivable. But bubonic plague has the lowest mortality rate. And then we have pneumonic and septicemic plague, and these both have much higher mortality rates when left untreated. Some research cites mortality rate as high as like 90%. Other sites cite mortality rate as low as 30 to 40%. Either way, though, these are much more serious diseases. With pneumonic plague, Yersinia pestis causes an infection of the lungs, and then this can occur through inhalation of an aerosolized droplet from someone that is already infected, and then that infection spread to their lungs, and then they released aerosolized droplets of this bacteria, and then someone else inhaled it. That is pneumonic. And then septicemic is like what we've been talking about, septicemia. This is when Yersinia pestis is directly introduced into the bloodstream through a cut or a wound, and it's going to be circulating through the body. And this leads to septicemia, or the active replication of this bacteria in the bloodstream, and a very extreme immune response. So you might be wondering why we're talking about the plague. You might be thinking the plague, this isn't the Dark Ages, but the plague is still very relevant today, and you might not have known that, but that is because this bacteria, Yersinia pestis, that causes the plague has so many intermediate hosts, and so it's very difficult to get rid of within a population. So it has all these different intermediate hosts from this flea infecting different types of animals. So it can infect many different types of rodents, like we talked about with rats and mice and squirrels and so on, but it can also infect large mammals like cows. Of course, it can infect humans, and it can infect a lot of things that are being kind of moved over long distance transport. So it's kind of moving throughout different parts of the world as well. So it's a very difficult disease to get rid of because it does have so many hosts. So we just saw with the plague that bacteria can be transmitted through fleas, but there are other types of zoonotic diseases that are transmitting these pathogenic bacteria that require these different arthropod vectors. So it can also be like lice and ticks too. An example of that is Rocky Mountain spotted fever, and this is caused by the bacteria Rickettsia rickettsii that's transmitted to humans from the bite of a tick. And this disease is endemic to North and South America, so it's relevant to us, but the incidence or the new cases of this happening is coincident with the arthropod vector range. So it's wherever this tick is present that you can potentially get this bacteria in your system. And so in terms of North America, that's most often going to be more mountainous and woody areas. So the incidence is very low in Florida, but it's still possible. It's going to lead to signs and symptoms like fever, headache, nausea, vomiting, and rash. And these are kind of very generalized signs and symptoms, so it is a little bit more difficult to diagnose. And that's why you have to be very aware of when you get a tick bite and what you start experiencing after having that tick bite, and sharing that with your doctor as well, because this is very serious. So it can disseminate throughout your body very quickly and be fatal in a very short amount of time, just within the first 8 days in healthy individuals, so people that aren't even immunocompromised. And so how can you prevent this? Of course, just be very aware of when you have a tick, remove ticks immediately, wash the area after you remove a tick, and protect yourself from areas that are more likely to have ticks by wearing like long socks and protecting your skin. Another example of a zoonotic disease that is transmitted by a tick is Lyme disease. And this tick is transmitting the bacteria Borrelia burgdorferi. And the tick that is transmitting that bacteria is the Ixodes tick. And these ticks tend to have a very complex life cycle with lots of different reservoirs, so they can use deer and mice and birds as part of their life cycle. So kind of like what we saw with the plague, it's difficult to get rid of this disease because their life cycle is so complex and they have all these different reservoirs. The symptoms of Lyme disease have 3 different stages. So there's this early localized stage where you see something called a bullseye rash in 70 to 80% of cases at the site of infection. Then you have the early disseminated stage and late stage. And these all have very general signs and symptoms, feeling like flu-like symptoms. But separate from what we just saw with Rocky Mountain spotted fever, though, Lyme disease fatality is very rare. So there's a very low incidence of mortality due to this disease, although those flu-like symptoms are also very serious as well, especially for people that are immunocompromised. Just a summary of some of the high points we just talked about and some of the big ideas. So bacterial infections of the circulatory system are very serious because the circulatory system is that fast-track ticket that just lets this infection now go anywhere in the body. So most often, circulatory system infections are going to have high mortality rates from that sepsis or bacteremia and that system-wide inflammatory response. Bacterial pathogens are going to require a breach of the immune defenses, so they're going to need help getting into your circulatory system. Oftentimes, we saw through many of these, there were arthropod vectors, or there were zoonotic cases with the bite or scratch of a cat, for example, or an open wound. It can also be through a hospital setting, like a nosocomial infection, the result of some kind of treatment or surgical procedure. For a lot of these, the symptoms are kind of vague and generalized, so they're difficult to target the exact thing that's causing that disease and a specific diagnosis. Most of these are going to be treated with antibiotic therapies. So we're talking about bacterial infections, and antibiotics are effective against bacteria. So most often they're going to be treated with antibiotic regimens if the bacteria is not resistant to that antibiotic. And then, of course, the systemic immune response to bacteremia involves a lot of release of these different cytokines and these different communication molecules that bacteria are releasing. And so sometimes that can be more detrimental than the actual infection. And that's kind of talking about septic shock. So septic shock is your immune system leading to that decrease in blood pressure because of that system-wide immune response, not so much from that bacteria releasing a bunch of toxins or anything like that. So sometimes your immune system and that exaggerated response from that bacteria just being everywhere at once is more detrimental than the actual bacteria itself. Now we'll talk about a few examples of viral infections of the cardiovascular and lymphatic systems. First, we have the Epstein-Barr virus, EBV. This is also known as human herpes virus 4. So it's a type of herpes virus. And it's associated with a variety of human diseases, and there are different types of herpes viruses that can infect humans. But human herpes virus 4 causes mononucleosis. And then eventually it could potentially lead to a cancer called Burkitt lymphoma from this initial infection and the transformation of those infected cells into malignant cancer cells, leading to Burkitt lymphoma. But this is not in every case, of course. This is very rare. And then nearly all people have actually been exposed to this virus at some time in their childhood. It's a very common virus to be exposed to, but whether or not that virus is going to actively replicate and kind of express itself is one of those factors that is not as well understood. Because since it is a herpes virus, it does lay dormant in cells, and then some external factor can induce that virus to start replicating and cause an active infection and infect other cells. So this virus is going to reside within B lymphocytes or B cells. So that's the type of cell that it is infecting. And like I said, since it's a herpes virus, it does remain dormant in a latent state for a long period of time until it's activated. And like I said, specifically with Burkitt lymphoma, it is associated with this human herpes virus 4 because these malignant cancer cells that are causing Burkitt lymphoma also have this human herpes virus in them. But it is not a direct relation or a one-to-one correlation between you have mononucleosis or you're infected with human herpes virus 4 and you will get Burkitt lymphoma. So that kind of transformation and progression to Burkitt lymphoma is not as well understood. There are also viral diseases of the circulatory and lymphatic systems that are transmitted by arthropod vectors. So oftentimes, again, these are going to be arboviruses that are transmitted by mosquitoes. And then the examples of these types of diseases would be chikungunya fever, yellow fever, and dengue fever. And really, the only thing I want you to take away from this is that these different types of diseases can be transmitted by mosquitoes, 1. And 2, look over here at these antimicrobial drugs. You'll see that there are no specific antimicrobials or antivirals for infection with these pathogens. And remember, that's just for a few reasons. So 1, viruses are difficult to develop drugs for because there are so few things that we can target and they're so closely related to the replication cycle of our cells. And then also, for the most part, these diseases are going to be self-limiting. So they can cause some really terrible signs and symptoms, but for the most part, in healthy individuals, these diseases are self-limiting and someone will get better just on their own. And then this one, though, in particular, yellow fever, does have a preventative vaccine that's available, but it's really only given to individuals that are at risk in certain regions where yellow fever is endemic. And then we've discussed this a few times already, but human immunodeficiency virus, or HIV, this is a retrovirus that over time can lead to acquired immunodeficiency syndrome, or AIDS. And that is when you have a low enough drop of the white blood cell count that it is considered AIDS. So there are two variants of HIV. There's HIV-1, and this is the worldwide or global HIV virus. And then HIV-2 is predominantly in West Africa, and it actually has a lower mortality rate, but is more easily transmissible. HIV is spread through direct contact with body fluids, so that would be sexual contact, use of contaminated needles, and so on. And then this specific virus is going to infect CD4 T cells, so your helper T cells. Remember, your helper T cells are extremely necessary for activating both your humoral and cell-mediated immune responses of your adaptive immune response. So over time, as these T cells decrease and you have a lower and lower helper T cell population in your body, you are not able to activate that adaptive immune response, which over time weakens your immune system to the point where you are not able to elicit an adaptive immune response. I thought this was an interesting micrograph showing how those HIV particles or viral particles bud off and leave that infected T cell. So this large blue thing would be the helper T cell, and then all these little green particles are the HIV, the virus that has replicated inside the cell and now is budding off of the surface of those helper T cells to go infect more helper T cells. And finally, we're going to talk about a few examples of some parasitic infections of the circulatory and lymphatic systems. First off, we have malaria. And malaria is the name of the disease, and what is causing malaria is infection with a protozoan parasite from the genus Plasmodium. Most often, that parasite is going to be Plasmodium falciparum, but it can also be other types of species within the genera Plasmodium as well. In the lab, you'll actually get to see Plasmodium falciparum in blood smears and be able to identify it in the microscope. So Plasmodium is going to infect red blood cells. That's part of its life cycle. And it also has an arthropod vector, so it has the Anopheles mosquito. So the mosquito is part of its life cycle. It's transmitting the infective stage to new hosts, and then we'll see on the next slide it's a very complicated life cycle, but part of the life cycle is actually infecting and replicating inside of red blood cells. So the classic signs and symptoms of malaria are going to really mimic the fever and chills of flu-like symptoms and signs. And this is a result of the bursting of those red blood cells, and it comes in cycles. So as that parasite infects more red blood cells and replicates and bursts, that happens over a series of days, and each time those red blood cells burst, you get those fever, chills, and flu-like symptoms. There is a vaccine now recommended by the World Health Organization since 2021, which is very cool. And the issue with this, though, is that it's very difficult to roll out in these regions where malaria is endemic. So it's been observed that it's a pretty effective vaccine, right? But it does require more doses over time. So having that dosage schedule and bringing that to a large enough population in endemic regions is proving a little bit difficult, but we are on our way to developing a vaccine for malaria, which is pretty neat. Here is the life cycle of Plasmodium falciparum, the causative agent of malaria. And this is not something that I will be asking you about on the exam. I just think parasitology is very interesting, especially when the mosquito does play such an important role in the life cycle of Plasmodium falciparum. It's not just a transmission role. It does play an important role for picking up these gametocytes and the actual fertilization of this parasite, and then the fertilized infective stage infecting the next human host. And then once this infective stage is in the human host, it actually has to make a first stop at the liver before it turns into the infective stage that is getting inside of red blood cells and causing those cycles of those flu-like symptoms as these blood cells burst. So a really interesting life cycle. But also, look at this complicated life cycle. What part of this life cycle are you going to target to reduce the incidence of this disease? Of course, you can target the mosquito. So you can protect yourself by wearing long sleeves, by putting up screens, by using bug repellent. So that's one way that the incidence of malaria is constantly being combated, by educating people on how to reduce the infection from the mosquito. Another example of a protozoan parasite that infects the circulatory system and uses the circulatory system to kind of move around and infect other parts of the body is Leishmania. And the infection of Leishmania, the disease it leads to, is Leishmaniasis. And this protozoan is transmitted again by an arthropod vector, the sandfly. So Leishmania is this protozoan, and it has some really neat virulence factors in order to avoid your immune system. So it does get phagocytized or eaten up by macrophages because your immune system sees it as foreign, but it has virulence factors to avoid being destroyed once it has been phagocytized. So it gets eaten up, and then it actually uses that macrophage as part of its life cycle. And the most common form of leishmaniasis is cutaneous leishmaniasis, and that forms an ulcer on your skin at the site of infection. But there are other forms of leishmaniasis. There's visceral leishmaniasis, and that's when this can move to other parts of your body, like your spleen and liver and bone marrow. Cutaneous leishmaniasis in and of itself is usually not treated. It's usually self-limiting, and it resolves on its own. That ulcer will resolve on its own over a period of weeks. But there are other forms of leishmaniasis that are more serious, especially in endemic regions. Like I mentioned with the spleen, liver, and bone marrow, there can also be cardiac involvement, and it can disrupt heart function. So it can potentially be a more serious infection, especially for people that are immunocompromised. But generally speaking, the most common form of the disease is self-limiting. So our last example for a parasitic blood infection or circulatory system infection is caused by species within the genus Schistosoma, and they cause the disease schistosomiasis. And these blood flukes are native or endemic to certain parts of the world, like the Caribbean, South America, the Middle East, Asia, and Africa. How this works is this blood fluke actually infects humans by penetrating the skin. And your skin will develop a rash or will be itchy where that penetration occurred. Or oftentimes people won't even know that they're infected for a long period of time. These flukes can live in the body for years. But infected humans shed these schistosoma eggs or cysts in the urine and feces because these flukes make a home in the hepatic vein or around your liver. And then they reproduce and shed these eggs or cysts through urine and feces, and then in fresh water that has been contaminated by that urine and feces, it finds its intermediate host, which are snails that actually live in that fresh water. A really interesting life cycle that we'll see on the next slide. So from the urine and fecal samples, you can diagnose schistosomiasis by observing it under the microscope and looking for the presence of cysts, or doing serologic testing to see if you've created antibodies against this certain type of pathogen. There are drugs that are available for this, so praziquantel is an effective treatment for this in particular. And of course, improving wastewater management and educating at-risk populations is a really important part of controlling the spread of this disease and lowering the incidence. Because in order for this blood fluke, this pathogen, to create or to finish its life cycle and actually create those pathogenic or infective forms of its life cycle, it has to get into freshwater habitats. So if you have clean fresh water and it's not being infected with things like urine and feces, then it's never going to be able to complete its life cycle, and then people will not be infected with the pathogen. So here's the life cycle of schistosoma species, and again, it's just really fascinating. So humans shed the eggs or cysts that these adult worms are creating and releasing inside of the body. These eggs then have to make contact with fresh water where snails exist, and it uses snails as part of its life cycle, its necessary life cycle, to develop into the free-swimming and infective stage. So it's this free-swimming infective stage that can penetrate the skin of humans and then make its way through your circulation to the hepatic vein, where it matures into the adult worm that will make cysts. just a really fascinating life cycle. And you can see here, though, that one way that we can kind of target this, one is understanding that wastewater management is very important, and having clean, fresh water access is very important. But two, targeting snails. Snails are the intermediate host, so if snails are not available in the environment, then this parasite cannot complete its life cycle.